So here inside our nucleus of a eukaryotic cell, we have our DNA. And our DNA is a helix or double-stranded. Now, our DNA is going to separate by an enzyme called RNA polymerase. And RNA polymerase will add RNA nucleotides to one strand of the DNA, and that strand that's transcribed into messenger RNA is called the template strand. The other strand, like in my picture here, the other strand of DNA that's purple, then just off to the side, is called the coding strand. It's untranscribed. This process here is called transcription. Now, if you're ever on a test or something, they give you a strand of DNA and they're asking you what's the corresponding RNA. This is what they're talking about. They're asking you, here's the DNA, now what's the RNA? And so this is transcription. Now you'll notice that RNA does not have any thymines. Anytime you think there should be a T, because that's how it is in DNA, you're going to put a U instead. Because RNA does not have thymines, it has uracil. It's a different kind of pyrimidine. And so, yeah, that's transcription. Now, the messenger RNA is going to leave the nucleus. Now, in us, in eukaryotes, we're going to have some RNA processing before this happens. A 5' cap and a poly A tail will be added, as well as introns will be cut out, and the exons will be spliced together. Now, once it enters into the nucleus, it's going to find a ribosome. Our DNA is still safe up there inside the nucleus. Now, our messenger RNA has a code that will tell the ribosome and the tRNAs which amino acids to bring. So tRNAs will bring the amino acids to the ribosome. They bring them to the ribosome, and ideally, or like really, there's just free-floating amino acids and tRNAs in the cytoplasm waiting to be used. So eventually, though, as the ribosome moves down the messenger RNA, there will be a growing chain or growing polypeptide of amino acids that could form up with other polypeptides or not to form a functional protein.